Gene therapy.
What is gene therapy? Gene therapy is when a DNA is introduced into a patient to treat a genetic disease. The new DNA contains a functioning gene to correct the effects of a disease-causing mutation. It is also an experimental technique that uses genes to treat or prevent disease. This technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery. How does gene therapy work? Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein if a mutated gene causes a necessary protein to be faulty or missing gene. Therapy may be able to introduce a normal copy of the gene to restore the function of the protein. A gene that is inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Viruses are often used as vectors in gene therapy because they can deliver new genes by infecting cells, and they are modified to prevent disease in humans. Retroviruses and adenoviruses insert their DNA into host cells' nuclei without integration. Vectors can be administered directly to tissues for gene therapy, resulting in functional protein expression. However, improving gene delivery, cell targeting, and gene regulation remains challenge. Types of gene therapy. Somatic gene therapy. Transfer of a section of DNA to any cell of the body that doesn't produce sperm or eggs. Germline gene therapy. Transfer of a section of DNA to cells that produces sperm or eggs. Categories of gene therapy. Somatic, cell gene therapy for the cure and prevention of diseases. Example, insertion of a DNA sequence into a person's cell to allow production of an enzyme like adenosine deaminase. Germline gene therapy for cure and prevention of diseases. Example, insertion of an adenosine deaminase sequence into early embryo or reproductive cells, which would affect not only the individual, but all of his or her offspring. Somatic cell enhancement. Example, insertion of a DNA sequence to improve memory, increase height, or increase intelligence which would affect only the individual. Germline enhancement. Example, insertion of a DNA sequence for enhancement into a blastocyst, sperm, or egg, which would affect future generation. Gene therapy techniques. Gene augmentation therapy. It is designed for diseases caused by loss of function of a gene. Introducing extra copies of a normal gene may increase the amount of normal gene product to a level where the normal phenotype is restored. Gene inhibition therapy. This is suitable for the treatment of infectious diseases, cancer, and inherited diseases caused by the inappropriate gene activity. The aim is to introduce a gene whose product either inhibits the expression of another gene or interferes with the activity of the product of another gene. Gene therapy killing of specific cells. Genes are directed to the target cells and then expressed so as to cause cell killing. Direct cell killing is possible if the inserted genes are expressed to produce a lethal toxin, suicide gene, or a gene encoding a prodrug is inserted, conferring susceptibility to killing by a subsequently administered drug. Summary. Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein. It aims to cure or treat genetic-related human illnesses. Somatic and germline gene therapy are the types of gene therapy. Gene therapy techniques include gene augmentation therapy, gene inhibition therapy, and gene therapy killing of specific cells. With some ethical issues on gene therapy, there are some pressing questions on whose authority or power to decide which human traits could be altered, and/or other concerns are on the discriminatory effects of those who may not or cannot avail gene therapy. Thank you for watching.